The premise of this study is that immune dysfunction exists in AML patients, and we noted in preclinical studies that in patients who received induction chemotherapy, particularly with a time sequential chemotherapy approach whereby cytotoxic chemotherapy agents are given over serial time points in order to prime leukemia cells for apoptosis. We found that there is an increase in immunosuppressive T cell subsets, particularly regulatory T cells, as well as significant T cell dysfunction at the time point when there's early recovery or early immune reconstitution after induction chemotherapy. So we hypothesized that if we can modulate these suppressive T cells at the time of early recovery after induction chemotherapy, that might lead to anti-leukemic clinical activity and perhaps synergize with cytotoxic chemotherapy. So we designed a phase 1 dose escalation study administering pomalidomide, which is an immunomodulatory agent. It's a class of drugs called IMiDs, immunomodulatory drugs. Pomalidomide is FDA approved for multiple myeloma. And pomalidomide has actually very diverse mechanism of action, but what pomalidomide particularly affects is a molecule known as cereblon. It targets cereblon, which is a receptor for the E3 ubiquitin ligase pathway. And by modulating cereblon, pomalidomide leads to the selective ubiquitination degradation of two transcription factors, iolose and Ikaros. And iolose and Ikaros are important regulators of interleukin-2 and overall T cell immunity. Pomalidomide leads to the degradation of these transcription factors, ultimately increasing interleukin-2 levels, and this can have significant modulatory effects on immune subsets, as we've seen in multiple myeloma and other cancers. So we were particularly interested in giving this agent during this early lymphocyte recovery time phase after induction chemotherapy, and we designed this phase 1 study whereby all patients received a timed sequential chemotherapy backbone of cytarabine, daunorubicin, followed by etoposide on days 8 through 10. And again, the premise of this is that leukemia cells would be primed to apoptose after each of these cytotoxic agents are administered. And pomalidomide was given in dose escalation at the time of early recovery with a median time point of 21 days after induction chemotherapy. This is the first study in AML with pomalidomide, and we escalated the dose up to 8 milligrams for 21 consecutive days, but noticed that there were 2 dose-limiting toxicities in this high dose level, and so we reduced the dose to 4 milligrams for 21 consecutive days and expanded on that cohort, and that was found to be the maximal tolerated dose. Main toxicities that we saw with the 4 milligram dose was rashes, which were self-limiting and able to be managed with supportive care. I think most importantly, we saw very encouraging clinical activity with the addition of pomalidomide to patients who received induction chemotherapy in newly diagnosed AML patients. We found a 77% complete remission rate in AML patients who received the time sequential chemotherapy followed by pomalidomide. And most importantly, we saw very encouraging clinical activity in poor risk subsets, such as those with unfavorable risk cytogenetics. We saw an 82% CR rate, and those with AML with MDS related changes, we saw an 85% CR rate. These are very poor risk subgroups that have very poor historical outcomes with conventional chemotherapy agents. Median overall survival on this study was 33.8 months, which again, small numbers, but compares favorably well to historical controls. I should mention that 43 patients received pomalidomide and the intensive chemotherapy backbone on this study. And ultimately, I think the correlates of this study were very interesting to us in that we found that pomalidomide significantly decreased iolose levels as a pharmacodynamic effect, and Aiolos is a repressor of interleukin-2, and so we expected that pomalidomide would decrease this transcription factor via its mechanism of action, and we found that mechanistically. And we also found that there were significant changes in the T-cell differentiation after administration of pomalidomide, which ultimately led to a decrease in suppressive and senescent T-cell subsets. So we were quite encouraged by the results of this phase 1 study, and I think this suggests that pomalidomide can modulate T-cell dysfunction after induction chemotherapy and may be able to abrogate the adverse risk biology of patients with unfavorable risk cytogenetics or those with AML with MDS-related changes. We're looking forward to designing and developing a phase 2 study, adding pomalidomide to intensive chemotherapy in patients with poor risk AML, newly diagnosed untreated AML, and hope that this can be a potential future option for patients in the clinic.